All right, it's now 11:01, so we'll get started with our webinar. Today we have a number of speakers from the Allen Institute, and today we'll be discussing the new Brain Knowledge platform and how you can use it in your own work. First, we'll start off with Tyler.
Hi, everyone. My name is Tyler Mollenkoff. I work in the technology group within brain science efforts at the Allen Institute, and I'm excited to get to share a number of things about what we've been doing, what's now publicly available, and where we're going with the Brain Knowledge platform. Within brain science at the Allen Institute, one of our main focuses is building a comprehensive atlas across all of these conditions that brings genes and cell types and brain function together in a cohesive way for the community to design better experiments, to gain insights just from looking at this data, and to build great models of brain function and health and disease. We highlighted in this slide, you can see how that spans species, modalities, space, and time. But over time, we find this set of conditions, sort of dimensions, grows, and the Institute's continuing to expand all the time across this. Next slide, please. So that's the mission scientifically, to build this atlas across all of those dimensions. What does the Brain Knowledge platform provide in terms of using that atlas? First and foremost, it's FAIR neuroscience data across all of these dimensions. If you're not familiar with FAIR, letters sometimes mean slightly different things, but findable, accessible, interoperable, and reusable, basically making the data really easy for you all to use and reuse and understand in conjunction. So we want to bring forward all of the data across these dimensions in one easy place. Second is discovery apps that are intuitive to use, so you can explore the connections between all of these aspects, between the anatomy and cell types, and gain new insights or design your better experiments. Third are machine learning and artificial intelligence powered tools for mapping new data into these frameworks, whether it's cell types or anatomy, and also making it easier for you to get started quickly, taking advantage of some of the amazing language capabilities coming out in the last few years. Fourth, lying on top of all of this data are data-driven knowledge in a few different dimensions. I think many of you are familiar with the Institute's anatomical atlases and the understanding of brain regions born out of originally ISH data and then more and more over time. But this data-driven knowledge expands to cell type, to precision genetic tools, and more. And we want to really focus on the data-driven side of this, particularly in an age where large language models can tell you lots of things, but they may not be grounded in really high quality and trusted information. And fifth is if your work doesn't take you to a website to do work, or you need to do things outside of it, great programmatic tools to take advantage of this cell type information, these atlases, and all of the related data. I'll walk through just a few visuals of what some of this looks like, and then we'll see some other demos and presentations of these things in action. Next slide, please. For comprehensive FAIR data, we've got a rich data catalog with a couple hundred different projects from the Allen Institute, from major initiatives like the Brain Initiative Cell Atlas Network, which is ongoing now within the Brain Initiative Cell Census Network, which preceded it, other foundations across the field, like Aligning Science Across Parkinson's, Seattle Alzheimer's disease work that the Allen Institute and others participate in. And for all of this, it's easy to search and find across many different dimensions. Raw data, or sort of really high volume data, is stored out at linked archives, many of them listed here. And you can see the provenance and access to papers for any of these data sets where that's available. Next slide, please. If you want to get into the data and make use of it, there's a number of different ways of discovering new insights from that data and understanding what it means and looks like. Our Allen Brain Cell Atlas has been around for a few years now and continues to get better and better. This lets you explore single cell information, either from single cell RNA-seq or spatial transcriptomics, and coming soon more modalities. with aligned information about cell types and brain regions and many more properties. NeuroGlancer is a wonderful open source community platform for working in 3 dimensions. And you can see here some visuals of 3D MRI templates, as well as new brain structures defined in some of our work on updating the atlases. And then for neuromorphology, we have some really amazing high resolution microscopy methods continuing to scale up and grow. And these full morphologies across the brain are available for exploration and search. Next slide. With ML AI powered tools, I'll highlight 2 here. One you may be familiar with is Map My Cells. This lets anybody in the community bring out their gene expression information from whatever assay, and really in some ways from any species. Submit that to the tool, and a couple minutes later, get information about the cell types present in their data based on these large community cell type references that are available in the Brain Knowledge platform. Several there across species and even across disease conditions available. On the ease of use side, we have a new AI assistant right now sitting alongside the ABC Atlas that makes it really lightning fast to get started with new visualizations and find the information you're looking for really very quickly. This is not a tool designed to tell you answers. We don't want to fall prey to hallucinations, but it's an amazing accelerator for letting you just describe simply what it is you're looking for and then see the rich data in the ABC Atlas very quickly. Next slide, please. The data-driven knowledge that's present in this knowledge base, I'll highlight across a few dimensions. And then you've heard me repeat these, you'll probably continue to hear those through the presentations. Cell types, anatomy, genetic tools. We have a number of different cell types taxonomies defined across different species, whole brain, parts of the brain, different conditions. You can browse all of those. They're backed by a large growing body of cells, over 30 million at this point. On the anatomy side, many of the atlases you're familiar with, all available there in a consistent sort of structure and format. And genetic tools, this is one of the newer data releases over the last couple of years from the Allen Institute, codified into this brain knowledge platform. So whether you want to target a specific brain region or cell type, you can get right into that. You can find the right genetic tool for your experiments. And for all of these, we want to present summary information about each individual cell type or brain structure or genetic tool. What does it do? Where does it come from? How should I understand it? And so these knowledge cards complement different views in ABC Atlas today, and we're going to continue to grow those. Next slide, please. And then on the programmatic side, we've got resources, again, for both cell types and anatomy, with tutorial notebooks for how to get started with the libraries we've made available, the data formats, the data schemas. If you need to work programmatically, there's some really great resources for getting into what the data is, how it's shaped, and how you can make use of it. I think we've done other webinars in the past to deep dive in those, and I think there's one coming up even in just a few weeks or maybe a couple months on to go further in that. Next slide, please. So a lot of these resources have been out there, but we've brought them all together in a really clear way in this Brain Knowledge platform. But this is still just the start for us, even though we've been these things have been developed over the last few years, and there's already a lot that's available. I'll highlight in this roadmap, kind of on the left, more data that we're working to release. In the middle, updates to some of these trusted knowledge references. And then on the right, more capabilities that are going to be coming so you can do more with these platform. And of course, we welcome any of your feedback and ideas. This roadmap's not set in stone. We've got a number of things in flight, but really want to hear from you all. What's most important and what would make the most impact in your work and in the field. In terms of more data, number of different methods within the realm of connectivity. Of course, understanding cell types and their distribution and anatomy is important, but how do the circuits work? How do these things connect to each other? Electron microscopy, barcoded sequencing, whole brain light sheet, and expansion light sheet microscopy. All of these are active areas of data generation by the Allen Institute and many other groups in the Brain Initiative. And the Brain Knowledge platform will be releasing these data in many waves over time. In development, particularly in the mouse, we've got gene expression and spatial data coming soon. You may have seen a large paper package about the developing brain. I think from the Brain Initiative Cell Census Network, some really amazing science in there. and we've got data releases planned around that. But scaling up to bigger brains, to human and non-human primate, there's a massive effort going on in the Brain Initiative to understand the cross-modality view of these data for spatial, single-cell, patch-seq, even connecting to some of the MRI information or functional MRI. So those data releases will be really exciting ways of moving from this high-resolution understanding we have now in the mouse to how that applies in the larger primate brains. For disease, there's already a number of Alzheimer's studies available, or Alzheimer's data sets we've got available, and some early Parkinson's information. We're going to be doing a lot more there, translating all of this great information in the healthy context into what changes in disease is really important to the Allen Institute's mission, maybe relevant to your work, and we're going to have a lot of data coming out in that regard. And then lastly, what does this all look like in the living animal? There's some amazing work in the neural dynamics group going on with in vivo physiology that can connect back to information about cell types and brain structures and other things. So some great stuff there. In terms of updating our references, we are on a path to update the Common Coordinate Framework, the adult mouse CCF, to a version 4. There are a couple parts of the brain that are sort of under-described, under-studied in CCF v3. There's some other ease of use things we want to do, and there's some new imaging methods that provide additional and alternative templates to map your data into this framework. So some really cool work planned for that. We also know that we'll have the whole brain understanding of cell types in the human, non-human primate mouse sort of consensus space. So as you're probably familiar, or in case you're not, over the last couple of years, there's been a really well-established, high-resolution understanding of all of the cell types in the mouse brain, over 5,000 of them, with multiple sort of atlas scale data sets aligning around that. We're stepping that up to a sort of multi-species shared understanding with human, macaque, marmoset, and mouse. A really exciting way to continue to refine a common language for all of us to speak around cell types. The same is true on the anatomical side. We've got CCF v4 for the adult mouse, but we are refining the brain structure sort of delineations and aligning the nomenclature for the human and non-human primate brains using community templates that are already out there and well established. I think it's going to be some great refinement coming out of work we're doing as part of the Brain Initiative. And then lastly, more capabilities, visualization and discovery. We could have a whole webinar about. There's lots going on there, but multimodality heat maps to get sort of even deeper views of the data, as well as differential search. We're going to be expanding that map my cells method from what is available today with gene expression to cell types, that you can bring in multiple modalities of data. You may have participated in one of the data challenges we ran along these lines. We know this is really important. As the first slide showed, understanding the brain requires so many different directions. We want to provide some very simple, lightweight tooling like Map My Cells today, but to help you come from whatever angle your work brings. We have a number of scalable processing pipelines the Allen Institute runs to make all of this available, to make our public resources even available to release in the first place. We'd love for the Brain Knowledge Platform to bring those out to you all so that you can run your data through those in cost-effective and trusted ways. We'll have many places we can continue to improve the programmatic tools. I could talk about that at length, but if that's of interest to you, please let us know and kind of what matters most to you. And ultimately, we want the Brain Knowledge Platform to be a place where you can not just work with data from the Allen Institute and broader Brain Initiative and community, but to analyze and disseminate your data as well. And that's a key part of the roadmap and where we're heading. So I think that's it for me. Thanks for joining the webinar today and for being interested in the Brain Knowledge Platform.
And we do have some couple minutes for Q&A with Tyler before he has to leave to another meeting. So if there are any questions or comments or suggestions about any of the topics that Tyler just discussed, please put that in the Q&A box in the Q&A chat function now, and we can answer some of those questions. And we have about 5 minutes or so to really cover questions based on these topics. And while we're waiting for some questions, Tyler, what are some of the things that you think are most exciting about everything that you just covered? I know you covered a breadth of things, but what stands out to you the most?
I think a cross-cutting theme is trying to bring resources and the community together. Um, and I won't say in one place because the field is too large and expansive for that. But I think the more that we can bring the community together, the more exciting things seem to happen. There, and we've started to see that some with the disease communities of how the Alzheimer's disease work at the Allen Institute and the Parkinson's disease work funneled through the Aligning Science Against Parkinson's. By looking at cell types in a shared space, you start to see some new insights about the sort of co-pathology and similarities in neurodegeneration generally. In groups like the Brain Initiative, we see the collaborative power of just getting people in the same room talking about these challenges in different ways, and we think the Brain Knowledge platform can help facilitate that, and it's really energizing for me anytime we get to see that in action.
Okay, it looks like we have a couple of questions. The first one that I see is, in addition to human mouse and non-human primate consensus type characterization, have you considered including rat, which is also a primary toxicological model?
Great question. You know, rat comes up a lot. There are, I think, some clear places we would like to be able to relate there on the anatomical atlasing side, but that's not, in our sights on the cell type atlasing side in the near term. However, if you know of, or if you can help, highlight active communities there where we can build technical and social bridges, I think that would be wonderful for the reasons you mentioned to tap into the uses for toxicology studies.
All right. And the next question that we have, are there videos that walk through some new users on how to use each of these tools? And I'll let Tyler answer, but I'll also be covering a bit of this in my portion of the talk as well.
What you want to take that one, Rachel? That's
Yeah, so we have developed some tools, some tutorials, some different PDFs and walkthroughs to help new users. And with the development of more tools and the addition of new atlases and new data sets, we will continue to create new tools and new helpful resources to walk through newbies as we keep adding new data sets and tools. So I can show where those are in my portion of the talk at the end. And another question that we have is, does Allen Institute focus on induced pluripotent stem cells derived from human brain organoids or assembloid models to study human brain?
great question. It's not an active area for us at the moment within the Allen Institute. We've got a number of collaborators we're working with who are very optimistic about the prospects for these kind of organoids and are doing some great work there. So we're really well tuned into how that's going and where these can be most effective in kind of in the experimental data generation paradigm overall. And yeah, so something we continue to want to learn about it and see how that can fit in.
All right, I think that finishes Tyler's portion of the webinar. We have no more questions for this first section. So next up, we can be joined by Sven, who will be going over the next portion of the talk.
Thank you, Rachel, and thank you, Tyler. Hello, everybody. My name is Sven Otto. I'm a product manager over on the data and technology side. And today I want to talk to you a bit about how the Brain Knowledge platform that Tyler just introduced relates to a platform that a lot of you are likely more familiar with, which is BrainMap, which is a platform that has been around for much longer than BKP has at this point. Next slide, please. Starting us off here, I really would like you all to think of BrainMap as kind of our parent web presence, if you will, that is centered around a lot of the scientific disciplines that Tyler alluded to in his introduction that we want to carry over into the Brain Knowledge platform. And that also serves as an access hub to access a lot of those historical atlases that have been around and well loved for the last couple of years. The mouse anatomy resources come to mind here, and then many of the other high resolution data sets that complement those atlases. The other nice thing about BrainMap is that it also offers an index, if you will, or additional front doors centered around those scientific consortia that Tyler mentioned, BICAN, so the Cell Atlas Network, BICCN, and others, as well as it's a great resource hub for tutorials, documentation, and similar resources that really enable scientists to use the resources that are available in BrainMap and in the BKP to their full extent. Complementary to BrainMap then, Brain Knowledge Platform, think of that as a product suite underneath the BrainMap umbrella. And in this case, we really want to move from these modalities more broadly to more specific scientific use cases to enable scientists to use these resources through a very particular lens. We'll start with things like doing experiments in your own lab with some of the genetic tools that are available from Allen Institute, all the way to gaining more insight from your own data through some of those cell type taxonomies that we have available through tools like MapMyCells. And I'll illustrate that a little further in a minute, as well as Alicia's demos will add further color here. The other nice thing about Brain Knowledge Platform is that we really want to embrace kind of an anatomy-centered exploration process. Scientists often are very focused on a particular region of the brain. And so we have added an anatomy front door that allows scientists to really explore this expansive data ecosystem through the lens of the ROI that they're interested in. And then last but not least, the Brain Knowledge Platform is also the ecosystem and the platform where we publish all of our latest data sets and the tooling to explore them. So with that said, I want to add a bit more visual to this text. So if you go to the next slide, Rachel, here's a quick video tutorial that I'm going to talk over. So you see here is the BrainMap homepage, and pretty much immediately you see the list of focus areas all the way from anatomy to hardware. These focus areas also have their own dedicated landing page. If you prefer a bit more of a focused view of these focus areas, where you can then explore them. The list of atlases that were mentioned, highlighting the mouse anatomy resources here again, that you can click through to access those historical resources that you're familiar with. The list of scientific consortia, again, allowing you to access a consortium-centered view of this data, the HMBA example being the latest one. And then in this case, I'm showcasing the documentation hub, where you can search both with free text search as well as faceted search filters across this documentation index, and can, for example, quickly and easily find the ABC Atlas user guide to learn more how to use that tool to its full potential. On this landing page, we are also featuring 2 different entry points to the Brain Knowledge Platform. And we are going to be clicking through to the BKP landing page here, where, again, high at the top, you see the scientific use cases that I was mentioning, starting with browse, experiment, exploration, analyzing, referencing, and developing, channeling back to the use cases that Tyler mentioned in his talk as well. And then most excitingly, in my opinion, we have this anatomy front door, where for now we have 3 species that you can select from: mouse, human, and in this case, macaque for non-human primate, where you are able to engage with these high-level brain regions informed by the CCFs, both in the side panel on the left as well as the visual on the right. And what this allows you to do is access pre-curated views of the data in the Brain Knowledge platform through the lens of these particular ROIs to get you started quickly on finding relevant to you information based on your brain region. And that's all for my part. I'm happy to take any questions before we dive into some of the end-to-end demos that Alicia will be showcasing for you. Thank you.
Yeah, if we have any questions for Sven, please put them in the Q&A box there. And there were a lot of changes to the website in the last couple months. Very exciting changes. I especially appreciate the search functions. I think it's a lot easier to find things now, and the way things are organized by data set and by all the different projects that are at the Allen Institute. We really hope that this new redesign and this new section with the Brain Knowledge platform makes it much easier for people to find and access all of these data sets that we have. And if you have any questions or suggestions for improvements or other thoughts, about this design, we really love to hear them as well.
Yeah, thanks for calling that out, Rachel. So for everybody's benefit, BrainMap received this major overhaul at the beginning of November last year, right before SFN. So if you've not had a chance to explore the new site or haven't visited BrainMap in a while, I definitely encourage you to check out the new site.
Okay, we have a first question for Sven. The question is, Sven mentioned in the newest data will be in the BKP. Will it eventually get incorporated into BrainMap as even newer data sets are published, or will there be different data existing in both places moving forward?
That's a great question. So again, I think of this as a kind of parent-child relationship. So all BKP data is also BrainMap data. That said, we will not be adding new data sets to the historical tooling that is available, just because technologies have changed over the years, and the BKP offers us more modern technologies to tap into for enabling exploration going forward. That said, both the more historical resources as well as the latest resources are intended to be complementary. And so we really see them as one big ecosystem, if you will.
And so, like with new transcriptomic data sets being added to the Allen Brain Cell Atlas, there have been, 4 or 5 data sets added to that over the last year or so. And I mean, that's still part of BrainMap, but technically that's also now the Brain Knowledge platform. So things have been continuously added to it, and things will be continuously added to it, especially based on the kind of data it is, right? Like data that fits, these large taxonomies will go in the ABC Atlas, right? You won't have currently that's the transcriptomic data is where that lives. Okay. Another question that we have is, I'm an individual engineer and I have an ORCID registration. What else do I need to get started? I intend to do AI LLMs research on ALS.
I think that sets you up pretty well, Lynn. So none of the functionality that is available today is behind a login wall, if you will. That may change over time, but for now, everything is freely available, and you can access all these open science resources right there and then.
Yes. So as Sven mentioned, there's no registration or anything like that or any login to use some of these data sets. We have some of the data is actually housed in larger data repositories. Many of them that are run through the National Institutes of Health. So NEMO, Dandy, some other ones that I'm thinking of. Some of our Alzheimer's data is online through the SAGE Alzheimer's disease portal, and some of the specific Alzheimer's data requires additional access through that particular portal, but that's for the unprocessed RNA sequencing data. So if you just want the processed data, we have access to that. But for almost all of our data, there's no login or portals or certain access points. But for a few of the data sets, depending on the project and the level of detail that the data contains, a lot of them are, again, housed also in external repositories that occasionally have their own access restrictions.
And there will be clear indications on the data collections that you may find for certain projects in the Brain Knowledge platform for whether a given data collection is open or controlled access to give you a better understanding from the start of whether you can just expect to download something straight away or may need to follow some of those additional administrative steps to gain access.
And for the vast majority of the data, especially shown in today's talk, that should just be able to be downloaded freely now. Like I mentioned, it's really some of the types, at least in our Alzheimer's study, that are a little more restricted access. But again, that just involves working with that external data portal and just submitting an access request for them. All right, another question that we have is, are there hands-on workshops provided on how to use the data on the BKP? Are these free for students, and how can we sign up for these workshops? Yes, we currently have 2 workshops that are scheduled in person at the Allen Institute in 2026 that are really aimed towards early career researchers, primarily graduate students. For one of those workshops, the application window actually just closed last week, unfortunately. But if you have students that have a strong computational background and are really interested in problem solving and creating new methods or creating new projects with the transcriptomic data on the BKP in particular, I would definitely recommend looking at the 2026 Computational Cell Types Workshop. And all of these workshops that we have are on our website, alleninstitute.org/events. And I can also type that in the chat here just so everyone has access to that. But we have a number of different workshops offered throughout the year. And again, I will just type that in the chat here, alleninstitute.org/events. And for some of our workshops, depending on which team is organizing them, some of them actually provide travel stipends for students. The workshops that I'm involved in particular, we offer some travel stipends. Some workshops have more limited funding, but the students will not have to ever pay to either submit an application or pay. There's no registration fees for these workshops. It's typically housing or travel that they have to pay for, or sometimes there are travel stipends associated. These are all great questions. Thank you. And addition to, we only have a limited number of these in-person workshops throughout the year, but we also have a number of online tools, tutorials, and sometimes we do virtual workshops as well with specific institutions. So if you have more questions about that, stay tuned for my portion of the talk.
Thank you all for those great questions. Appreciate your interest into the Brain Knowledge platform, and I'll hand it over to Alicia.
All right, now we can move on to Alicia's portion.
All right. My name is Alicia Fiamani. I am a product manager with the Allen Institute, working closely with Sven and Tyler. I'm going to go a little bit deeper into some of the tools that you've already seen today and show you kind of how they're all connected and how they can be used together. So I've got a short video similar to what Sven showed, where I'll be talking over it to give you a sense for how to progress through the tools that are available on the Brain Knowledge platform. Go ahead and get started. So I'm going to pick up where Sven left off with our anatomical navigation. So start with your region of interest. Here we're going to pick the striatum. You'll click there and you can access a number of resources. Clicking those links will bring you to the resource filtered by that area, that region of interest that we selected. So this is taking us into the ABC Atlas with a pre-filtered view to the striatum. So you'll see here that we've got 2 data sets that are loading, one spatial, one 10x data set. you'll see that they are populating with dots. Each dot represents a cell. And as I hover over, I'm seeing a little bit more information about those cells. Let's say I wanted to get a high-level overview of what I'm looking at. This that you're seeing here on the left is our new assistant. This is an LLM, and you can ask it questions such as, what am I looking at? The assistant is able to access the view that is displayed here in the ABC Atlas, check the metadata, and give you more information about the data sets that you're looking at. And so here you'll see that it describes these two data sets, and it says that it's being colored by the cell types. And so I'm interested in, or filtering by anatomical region. I want to filter, say, by cholinergic cells. The assistant is able to interpret this question and kind of come up with a suggestion. So it's found a neurotransmitter type. In this case, I actually want to see cell types. I want to see the cell type taxonomies that are related to cholinergic cells. So I'm able to refine my request based on this feedback. And it's come up with a few cell types that might be relevant. So we're going to go with this first one, the Palstratum gabacholinergic cells. And so I want to filter this view down to that particular cell type. It's asking me if I'm sure. And then it's going to actually manipulate the view. So not only can you ask it questions, but you can also ask it to do things, to show you things, and to apply filters to open data sets. There's a lot of things that it can do based on the data that it has access to in the ABC Atlas. So here, you'll see that it's filtered those cells down further, and I've been able to get down to a subset of cells that I'm interested in learning more about. In this case, I'm going to ask it how to download this data. It's going to respond with a link to the ABC Atlas Access GitHub page. So this is where you can go to download all of the data that's available in the ABC Atlas. We have tutorial notebooks, as Tyler mentioned before, that guide you through all of the data and metadata, and there's tutorials on how to create your notebooks to access the data in the way that you want to. So in this case, I can use these notebooks to construct a query to access just the subset of cells that I was looking at in the ABC Atlas. So I encourage you to explore this site. Even if you're not an expert in Python, the notebooks are designed to walk you through. Now let's say that you want to design an experiment based on this cell type. Our Genetic Tools Atlas offers a way to filter down the tools that are available based on a number of categories. In this case, I'm going to filter by the region of interest that we were looking at, the striatum. Apply that, and it will filter down the list. And filter further by that cell type that we were looking at before. can do further filtering. So, for example, we have our Hall of Fame cells, which are tools that are proven and high quality. And I can apply additional filters here as well. And you'll see now I have a set of tools that are relevant to that set of cells that I was looking at before. I want to go even further and look at high resolution images of the cells that would be targeted. I can do that. This is opening a tool called NeuroGlancer that's available to allow you to really go deep and see a really high resolution image of the targeted cells here. I just think these are really pretty to look at as well. You can see there's controls and tools here in that visualization tool as well. And from here, if you wanted to learn more about acquiring those tools, you can jump right into Add Gene. So that's a high-level overview of how all of these tools are linked together. I'm going to go into a couple of them in a little bit more detail and give you examples of how you can learn more about cell types and all of the data that's available in the Brain Knowledge platform. Next slide, please. All right, so you saw in the ABC Atlas how you can hover over a cell that's displayed, and you'll see in the screenshot there that I'm hovering over it and has a box that pops up that tells me what cell type it is. It'll actually show you information based on the color that you selected. In this case, we're coloring by cell type, so it's showing me the cell type that's highlighted. You also see two additional buttons there, a Learn More and Remove Filter. So if we click the Learn More, it'll show you this tab on this panel on the right here that explains what the acronyms mean. It goes into detail. In a lot of cases, the nomenclature can tell you a little bit about how that cell type was defined and what is known about the cell type. Excuse me. Next slide. In addition, you'll see there's two tabs there. We were looking at the Overview tab before. The Marker Genes tab shows you which marker genes were involved in defining this cell type. And if you click on the Add Genes to add the set to the genes, you'll be able to color your existing visualization by the genes that are defining this cell type. So you can see how these cell types are defined by their genetic profiles. A little bit more about the Genetic Tools Atlas. Really excited about this. It's something that Sven has worked deeply on. So if you have questions, he can certainly answer them. But we are excited because these are tools available to the community to help you use these cell types to design your experiments more precisely. And the tool that this Atlas tool helps you really target specifically those cell types and areas that you are interested in with a lot of precision. So let us know if there's things you want to see here. We're always interested in hearing from you. Next slide, please. Yeah, and here's an example of how you can target specific cells for relevant experiments. Going to the next slide, you mentioned here, Map My Cells. This is a great tool if you have your own data. So Tyler mentioned this earlier. If you have your own transcriptomic data, you can actually apply the cell type labeling using MapMyCell. So you upload your own cell by gene data, and you'll see it's a little bit small in this slide, but you'll get back a file that shows you these same cell type labels that you saw in ABC Atlas for your own cells. So you can then go into the ABC Atlas, look up that cell type, learn more about how it's defined and the marker genes, and access all of those tools and resources that are connected to those cell types. So really giving you a 360-degree access to the data that's driving these cell types, and the research that's being done here at the Allen Institute. And with that, I'll hand it back over to Rachel.
Do we have any questions we had some questions in the chat about navigating and finding some things. I responded to most of them, but as Sven had mentioned previously, our website went through a bit of an overhaul the last couple months. But anything that we've talked about today on the Brain Knowledge platform, you should be able to find through brain-map.org/BKP. Or now that we have that nice search function, you should be able to just type in that search bar, specific things that you're looking for. But if there are any more questions related to some of the things that Alicia has shown, we have a couple minutes to go through that. And yes, Sven just put, here, I'll put this in the chat. This is a message from Sven. This is showing that, just highlighting the specific cholinergic cells, the genetic tools that Alicia highlighted in her demo. The actual link to those specific tools and those cells are now in that link in the chat that was just posted. And again, there's a lot of information all over on our website, and we do understand that it can be difficult to find things. So please reach out to us, let us know if you're having difficulty finding things. All right, and with that, we can move on to the last portion of the webinar. I'm Rachel Hostetler. Unlike the other three presenters, I'm not on the data and technology team, actually, so I'm not involved in the tool development, but I'm more working on the scientist side, and I work with outreach and dissemination of our data and tools. So I get to work very closely with members of our data and technology team who are involved in making these great tools, and my job is to bring it to others, to the masses. And so how do we disseminate some of these tools? So in particular, through the grant that I am on through NINDS, I'm on a U24 grant that is very much focused on data dissemination. And so this has been brought up a couple times throughout the talk. How are we doing workshops? How are we teaching people to use these tools? Over the last 2 years or so, we've had a number of workshops either hosted in person at the Allen Institute or hosted at conferences. So usually at SFN every year, we have an event. If you're in Europe, we plan to be at the FENS conference this summer, 2026. In Barcelona, we'll have a table there where we can discuss and help you use the tools. If you're an Alzheimer's researcher, we will also be at the Alzheimer's Association International Conference this summer as well in London. So those are just some of the ways where there are opportunities to interact with members of the Allen Institute in person and perhaps have the opportunity to get more one-on-one help using some of these tools. So we've also done a number of in-person and virtual seminars, primarily at universities, so that we've done some talks with graduate students, postdocs, and faculty. And if that is something that you are interested in, feel free to email me. I can put my email in the chat. It is rachel.hostetler@alleninstitute.org. So again, part of my position is running some of these seminars and virtual or in-person events with different universities. and this webinar series. So we've been doing this webinar series for about 2 years now. This is, I believe, our 16th webinar, and we've had tens of thousands of views on these webinars, and we will continue making these webinars. Next month, we will have a webinar specifically focused on the programmatic access to the transcriptomic data in the Allen Brain Cell Atlas. And then in March, we will have a webinar focused on the new consensus taxonomy for spinal cord transcriptomic data. And again, all of these webinars are also available at alleninstitute.org/events. So if you found the registration page for this webinar, it's the same registration page for those other webinars as well. All right, and we've also created a number of different tutorials. We've made some video tutorials. Some of these are embedded into the actual website. We've also made some written use cases to help people understand and navigate some of these tools. And so these are just some of the different ways that we're creating these different guides to really help new users walk through and really use some of these tools and figure out how they can use them in their own lab or for their own research or for teaching. We have a number of documents and resources specifically for educators. So if you're teaching an undergraduate class, teaching even perhaps even a high school level class or a graduate level class, we have a number of different educator resources as well that help you bring some of these large scale data sets into your classroom specifically for teaching purposes. Some other ways that we're supporting our external users. So again, we've been providing hands-on support, really doing office hours at a lot of these conferences that we go to. We also have some virtual office hours set up as well, which again will be featured on our events page. And we've also been doing some one-on-one support. So I put my email in the chat. So if you're using some of these tools and you're reaching some dead ends or you have some questions or something's unclear, please feel free to email me because part of my position also involves providing one-on-one support to scientists who are trying to use our tools. And we've also created a new centralized resource support page, which is called Cell Type Support. And this is really where we're trying to centralize all of our tutorials, protocols, and other things related to categorizing and profiling cell types. And so that's listed here at brain-map.org/support/cell type support. So that blue link there. And in addition to disseminating these data, providing these tutorials and tools, another reason why it is so critical and important for us to interact with users of our tools is to also get their feedback, hear their voices. And so when some of these tools were released, maybe a year or two ago, there was some common user feedback. Like, I don't know what cell type XYZ is. I don't really know what these acronyms are. I want to know where the marker genes are for these cell types. How do I access the programmatic data? Can these tools do computations? These are just some things that we have heard and gotten, feedback that we've gotten from users. And so Alicia already showed this in her section, but I really just want to highlight this again. So about a year or two ago, when the Allen Brain Cell Atlas or ABC Atlas was first released, there really was not too much information describing all of the cell type names listed there on the left. So we have things like OBCR GLUT, we have things like DGIMN GLUT, and for those familiar with certain cell types, those acronyms might make sense. But then once you got deeper and deeper into the taxonomy, the acronyms became a bit more complicated. So as Alicia mentioned, after repeatedly hearing this feedback from our users, these new panels were created and are now integrated in the actual tool itself. So there's a little Learn More button that appears on the left-hand side in purple. And if you click on that, it actually pops out this panel, which gives you very detailed information about the actual nomenclature used for that cell type. And there's a separate panel for marker genes. And this was, again, after hearing feedback about, this tool is difficult to navigate. I'm not sure what these things mean. I'm looking for this information. How do I find it? So again, it's very, very important through webinars like this, through our workshops, and through other avenues to hear feedback and hear things that could be improved with the tools. And just some additional links to highlight. I've mentioned some of these already, but again, all these webinars and all of our workshops are at alleninstitute.org/events. That cell type support resource page that I mentioned is listed up here in the upper left-hand corner. We also have a community forum. So if you have not used our community forum, I highly recommend logging into that. So the community forum is where you log in, you create a username, and this forum has been running for 6 or 7 years now at this point. So there's many years of information and questions that people have asked. And then the scientists here at the Allen Institute, we go through the questions, we triage them, questions to certain people, and we do our best to answer all of the questions that people post in there. And sometimes people just like to post questions, understanding a certain tool, or maybe they're having some difficult results. For example, if you use the Map My Cells tool, which Alicia highlighted, right? Map My Cells is a tool where you can, if you have your own transcriptomic data, single cell or single nucleus or spatial, you can map it to one of our transcriptomic taxonomies that we have at the Allen Institute and then get those cell labels. And sometimes the mapping doesn't work correctly. So if you have an issue with that mapping, you can post about it in the community forum. And Scott Daniel, who is one of our software engineers who runs Map My Cells, he will get back to you as quickly as he can, and he's pretty quick, providing very direct answers and support and trying to work through the code or if there was any bug that went through with your mapping. So community.brain-map.org is a very useful resource. All right, and with that, that is the end of this webinar, but we do have a few times for Q&A, and I also do very briefly want to launch a poll real quick, and in this poll, it will take a couple of seconds, it should appear on your screen, and this poll is just very helpful for us designing webinars going forward, and it's also helpful to hear feedback about the tool. So if there were certain tools or things that you liked or had questions about or want to learn more about, you can please fill out this poll and submit some of your comments and feedback there. And we also have a few more moments for questions as well. So if there are any other unanswered questions, please submit those in the Q&A box.
Rachel, I may have accidentally closed a question when I submitted the text answer to it. So if you scroll down, there was one question around mapping the whole mouse brain. Is there any other resources that you or Alicia can think of there for it besides Map My Cells?
Yeah, I see the question now. Yeah, I see your answer. Yeah, I think if I want to register and map the whole mouse brain, is there any specific tool for that? So yeah, if you have transcriptomic data, that's where Map My Cells would be used. If they're thinking of like mapping or registering imaging data, like mapping to the CCF, that would be another process. Yeah, so I think, yeah, if what you're mapping is transcriptomic data, then yeah, Map My Cells would be the best way to do that. Imaging data. Okay. Yeah, so I think I'd have to double check and look at or the original in situ hybridization atlas. I assume that would have CFOS in there. So if you have like immunostaining and you're trying to map it to the atlas. I think, yeah, registering to the CCF would probably be the easiest way. And we have a number of tutorials on how to do that on our website. So CCF, the Common Coordinate Framework, would be one thing to search for. And also searching through that community forum that I mentioned, there could be someone who's already done this mapping, and that information could be available in the community forum as well.
Didn't we also have the Map My Sections data challenge, Rachel, that was somewhat targeted at this use case?
Yes, yeah. So we just had a data challenge that was specifically aimed at taking spatial transcriptomic data and sort of mapping it to the genetic tools data. So we don't have any official tool that does that right now, but we did just recently host a data challenge where we challenge the community to try to come up with mapping algorithms to address that issue. Okay, well, thank you very much for, let's see, okay, one more question. Okay, and the data challenge that Sven just mentioned, I just posted that in the chat as well. Again, that's not any tools that we designed, but there are links to the tools that are data challenge participants designed, which might be useful for particular use cases as well. All right, well, thank you everyone for viewing this webinar today and for participating in this poll. Let's see, another quick question that we have is, do you also have plans to bring some transgenic mice for some diseases? I do want to say that there are a couple different gene therapy projects going on at the Allen Institute right now, which one of them is for Dravet syndrome, which is a seizure disorder, and there are a few other ones as well. But the transgenic mice and the disease model mice that we have is really aimed towards a gene therapy aim. And let's see, I can link that in the chat for this as well. Just a moment. Here.
There are a few transgenic lines that are also featured in the genetic tools atlas already. However, I'm not sure how disease-centered they are.
Yeah, because most of our because like we don't have, like, all of our Alzheimer's work is done with postmortem human tissue, so like we don't have an Alzheimer's model for mouse that we're working with. So most of our mouse work is done in Black 6 mice, and most of them are not genetic models. But if you go online to the genetic tools link that I put in there in the chat, that also includes a list of all of the different transgenic mouse lines that we have as well. So a lot of them are Cree lines and different kinds of driver lines. So anything that you're looking for that you're interested in, you should be able to see if we have it or not through there. Okay, all right, thank you very much. That is all the time that we have for today's webinar. And thank you again for all of your questions and feedback. It's really helpful for us as scientists and as tool developers. All right, thank you, everyone.
Thank you.